Still. I'm the assistant student coordinator for Project SEED. I work at Franklin Central High School where I teach ICT and AP Chemistry, and I actually got involved with Project SEED through Deb Dickerson, who teaches chemistry there, and actually coordinated Project SEED for many years. I'm really grateful to have this opportunity to work with Project SEED. The kids are absolutely amazing, and the fact that they get to do this real research that most people don't have the chance to do until undergraduate or even graduate school is just absolutely amazing. I've enjoyed, getting to know the kids, getting to have picnics with them, play frisbee with them, go for walks along the canal with them. And that kind of goes along with some of the things that I like to do. In my spare time, I like to run, work out, ride a horseback ride when I can find a horse. I tap dance, I fence, I scrapbook. But mostly I've just enjoyed having something to do this summer rather than sitting around and doing nothing. And these kids are absolutely amazing, and everything that I want to thank everybody for making it possible. The fact that we have 24 students doing actual research around IUPUI and at various other places around, it's an opportunity that these kids couldn't have anywhere else. So thank you very much for giving all your time and your money and your talents to the project. And I look forward to many more years with the program and helping more students become involved and be able to achieve their dreams.
Hi, I'm John DeBroda, and I've been working with Dr. Horia Petraki and also with Dr. Bruce Ray on a protein called alpha-1 antitrypsin, which is abbreviated A1AT. We're most interested in the interactions with biological membranes, and we're simulating those with lipids. So there are many different reasons why we want to study A1AT. It is found in the lungs, and if there's a deficiency in it, then it leads to diseases such as emphysema, which is where lung tissue breaks down. Also, cigarette smoke leads to emphysema, so we think there might be some link between that and A1AT deficiency. I'm doing different kinds of experiments and learning different methods to prepare A1AT and lipid vesicles for study. One of them is lipid extrusion, where you pass the lipids through a small cross-sectional area of holes, and it breaks multi-layered laminar vesicles into small unilaminar vesicles where it's just one circle and they're really small. You do that by pushing syringes with the lipid back and forth across the membrane. Another type of method that I've been doing is NMR spectroscopy, which is nuclear magnetic resonance. And the way that that works is it takes advantage of the spin that protons have. Especially it can show really small molecular changes. So what we've been doing is placing the A1AT alone in the NMR machine, placing the lipid, which is called DOPC, alone in the NMR machine, and also placing them together and seeing how those things changed so that we can observe the interactions between the lipid and the protein. Finally, I've been working with UV spectroscopy, which is where it measures the absorbance of photons of light at changing wavelengths, starting at about 500 nanometers all the way down to about 240 nanometers, which is mostly within the ultraviolet spectrum. The absorbance of a substance changes based on the substance and the properties of the substance at the given time. So we've changed things like temperature and done a spectrum from about 20 degrees Celsius all the way up to about 70 degrees Celsius and seen how that changes its absorbency across the spectrum. We've also done some chemical denaturing with urea, and that also created differences that we can use to compare to when we compare the lipid and the protein together, which has shown that we're not exactly sure what the interactions are, but there's definitely something happening, which is exciting. Future projects, I saw in the Med Center some gel electrophoresis, and that looked like something interesting that we might be able to do with A1AT. And also, perhaps before I leave here, we'll be doing some X-ray scattering with A1AT as well.
Hi, my name is Emily Lucas. I'm in the School of Medicine in the Department of Microbiology and Immunology. I'm working with the bacteria Haemophilus ducreyi and Haemophilus influenzae. Haemophilus ducreyi causes the genital ulcer disease chancroid, and chancroid is a major public health problem in underdeveloped countries, and it facilitates the transmission acquisition of HIV/AIDS. Haemophilus ducrei and Haemophilus influenzae are human pathogens, and part of the body's defense, it secretes antimicrobial peptides. But these antimicrobial peptides don't kill Haemophilus ducrei and Haemophilus influenzae, so we're examining the mechanism that allows them to survive. And right now we don't have any results, but we're expecting them in about 2 days, so hopefully that'll be done by the poster session. So to study the mechanism that allows these pathogens to survive in antimicrobial peptides, we perform AP assays, which is basically putting the bacteria in different concentrations of the peptides and then studying survival and the growth.
Hi, I'm Philip Sanders and I work in the lab for bioinformatics. My project is computational analysis of biological networks. My preceptor is Dr. Matthew Palikal, and I work with Deepta Lijam, who helps me with the biology part of my project. This project, what I have done is based on processing biological data. What it does is we take networks that are created with interactions between proteins and we analyze them and find the important parts of the network to be able to show biologists where to look for when they're searching for various biomarkers and searching to cure diseases, or in our case, when they're researching. My job is to create a program that takes one of these networks and finds various network statistics on each of the individual nodes, a better idea of what proteins we're looking at here and what their functions are. Ordinarily, with a small network, it would be easy to find the important nodes, but we're looking at networks like this. Now, I don't know about you, but I can't make much sense of this network, and that's where my program comes in. I started here not knowing much about programming, having a very basic base in it. But as I came here, I researched it myself, and I was able to learn more about programming so I could make my program. Then, after I made my program, I would take small sample networks, put it through my program, and make sure all my statistics were working right. Then, I moved on to a much larger, real biological network. Now, this biological was taken from proteins found in a salamander that regenerates its limbs. They cut off the arm of the salamander and saw all the proteins that were interacting there. Then, using these proteins and another program, they made a network of this data. I took my program and I ran this network matrix through my program, and I came out with various statistics, various data. Now, these statistics will say this protein is central to the network. That's all they say. But we can assign a biological significance to these statistics that will tell biologists that instead of this protein simply being central to the network, this protein plays a regulatory part. It regulates other proteins. Or this protein makes a functional module. Using this information, biologists can be better oriented into directing their research, and that's my project. And the goal of this project that I'm working on is to allow humans to regenerate their limbs. There's too much biological data for biologists to go through it by ordinary means or by simply looking at it. They need programs like mine to be able to sort through all this stuff, to really understand what they're looking at.
Hi, my name is Maria Alatorre, and I'm working at the VA Medical Center with Dr. Jairam and Dr. Padma. We are studying chemogene therapy for colon cancer. Colon cancer is the second leading cause of death in the United States, so the work in this lab is very fundamental. There is a drug called tiazofurin we used for treating colorectal cancer, but it's not very effective. It's only about 20 to 30% effective. The drug works by converting tiazofurin to TAD with NMNAT2. TAD is an analog of NAD, and so it'll compete for a site on IMPDH. This will inhibit IMPDH, which will inhibit the production of GTP and DGTP levels in the cell. GTP and DGTP are responsible for nucleic acid, so RNA and DNA synthesis, and signal transduction, processes that are vital to the cell. So the main steps in this experiment are construction of the vector with the gene of interest, plasmid preparation, bacterial transfection, harvesting and purification, viral titer termination. So the first step, construction of the vector, that's where the gene of interest, HMNAT2, is inserted into the adeno-associated virus, into the multiple controlling site. Once the vector is constructed, we'll use two other plasmids from the AAV helper-free system, plasmid adeno-associated virus RC and plasmid adeno-associated virus helper. And together, the three plasmids will have all the genes necessary for the generation of the virus. The second step is bacterial transfection, and this is where each plasmid of the AAV helper-free system is put separately into the bacteria, and this is done through heat shock, and this is to have a lot of the plasmid, so multiplication of the plasmid. Next step is plasmid preparation, and this is where we extract the plasmid DNA from the bacteria. This is large-scale plasmid preparation. Then we take the plasmid and from the plasmid DNA from all three plasmids, so the PAV RC, the PAV helper, and the adeno-associated virus with the gene of interest. We'll take all three and we'll put it into HEK293 cells, which is human embryonic kidney cells, and this is called triple transfection. Leave it in there for 72 hours, and during the 72 hours, all the plasmids will generate into the virus with the gene of interest, HNMNAT2. Then we harvest the cells, so we collect the cells, and then through 3 to 4 cycles of freezing, thawing, and vortexing, we'll extract the constructed virus from the kidney cells. The next step is viral purification, and this is where we extract just the virus, and all the other cellular debris is taken away, and so we get just the virus. Finally, this virus with the gene of interest we can use to transfect colorectal cancer, but right now we're instead of using a vector with an HNMNAT2, we're using the vector with GFP, and that's green fluorescence protein. So to know if the generation of the virus really did happen, we'll see through the microscope if for little green fluorescent spots, and that means the virus did generate. But we'll do that first before doing HNMNAT2, so we know it works.